Presentation. So Gene and I are going to team tag, as Barton mentioned, and I'll just try to set the stage with how do we think about some of the nutritive or forage quality constituents in hays, haylages, pasture. I'm probably going to be focusing more on hay and haylage in this talk, but let's talk about pasture as we go. And then Gene is going to follow through with the interpretations and how do you make decisions about allocating all these wildly different resources, forage resources that we have. Even as really good managers, we always have tremendous variation in our forage resources. It's part of the system we manage. So we have to make decisions about what do we do with the higher value, the lower value forages to use them to best advantage. So I've already given you some of that in the overview. I'm going to give you a brief definition of what do we actually mean by nutritive value or forage quality. Those are interchangeable terms in my opinion. I want to talk about a very simple but powerful way you can think about how the dry matter in your forages, let's exclude the water, let's ignore the water, just the dry matter that animals are consuming. There's a very basic division of that dry matter into a portion that's virtually 100% digestible. We call that cell contents. And there's a portion we call fiber that's partially digestible. I want you to just get some sense of, well, how do I kind of grasp what those levels might be in my forage and what does that mean for my animals. We'll talk a little bit about plant species, plant parts, meaning leaves or stems, maturity stages and how those affect quality. I think most of you already know that. Forage sampling approaches and methods along the same lines that Lewis covered. How do you go about selecting a forage testing lab? We don't have a state-specific forage testing lab. You can send your samples anywhere. And maybe that's one way in which we're a little different in forage testing than in soil testing is you have every reason to think you should get the same results from multiple labs, whether it's California, New York, Wisconsin, Georgia. Do we get similar numbers? No. It can be just as confusing as the reports Lewis talked about that come back from Penn State versus West Virginia. So talk about that a little bit. And then Gene's going to cover the last part here. So definition, my simple seat of the pants agronomist definition of nutritive value or forage quality is the level of animal response, animal performance. So weight gain, milk production, wool production, draft animal pulling power, however you want to think of animal performance in response to some forage source that they're fed, as long as they have more than enough of it to eat. So you're not measuring a response to quantity, you're measuring a response to the composition of that forage. So in other words, they're not limited in the amount they can eat. That's part of the definition. Ultimate test, of course, is animal response. We'd really like to know what is the average daily gain going to be on this particular truckload of hay or this particular field of haylage that I put up. It's, of course, time-consuming, expensive, takes an awful lot of forage to do that, and we only learn it after the fact. It's too late. So we have to think about alternative methods, laboratory methods that give us estimates that we can turn into animal performance estimates. We use forage testing to define some of the specific feed constituents, forage constituents, dry matter constituents that we have particular interest in, like if we think we have to supplement protein or we think we have to supplement energy. We can just focus on those particular fractions, constituents. We can hopefully predict animal response and even more importantly, economic returns to our decision making. And then if we anticipate that there might be an anti-quality problem, like maybe nitrates or possibly endophytes from infected tall fescue Those are some specific tests that we don't normally do, but they're on the list, and we could have them performed for very reasonable prices. I'm probably not going to talk about those, but we could bring them up in questions. And then in terms of the marketplace, if we're marketing, either buying or selling, an awful lot of hay, at least, is sold on the basis of protein or fiber. MDF is neutral detergent fiber. A lot of it's sold on the basis of either relative feed value or relative forage quality. I'm not going to really go into those, but those flow very easily from our conventional forage tests. These are calculated values that come from fiber fractions. Okay, so just to kind of set the stage, we're talking about species, we're talking about plant parts. I'm just going to talk in very general terms about the digestibility of the overall dry matter of these different plants and parts in a ruminant. Sheep, cattle, goats, musk oxen, bison, deer, elk, whatever. So let's look first at just the legumes here. We've got an alfalfa, we've got a white clover. And let's think about within legumes, what's happening with stems versus leaves? What's the relative digestibility of these different parts? Does it matter which parts they're eating? So if you look at stems, in general, we're somewhere in the neighborhood of 50 to 60% digestibility in the stems. What do you think the leaves are going to be? Higher? The same? What do you think? Higher, of course. So leaves are going to be 70 to even 85% digestible. So obviously, the most valuable part of the legume plant in terms of energy is going to be the leaves. Okay, no big news there. Let's compare with grasses. So if we look at stems of grasses, 60 to 75% digestible. That's actually higher than it was in legumes. That's kind of interesting. You normally think of grasses as maybe being lower nutritional value than legumes, but look at that. The stems are actually more digestible in grasses than they are in legumes. This is generalities. We can find exceptions. Let's look at the leaves. Leaves aren't quite as digestible. They're not far behind. They're almost as digestible in grasses as they are in legumes. So I want you to reconsider some of the thumb rules we've come up with over the years that tend to penalize grasses as being lower digestibility, lower quality, because a lot of that comes from old-fashioned, outmoded forage testing that we've moved away from. It left a legacy of this opinion or concept that perhaps grasses are not as valuable to us, and that's changed as we've implemented more modern forage testing. All right, if you take either of those, the grass or the legume, immature, moderately mature, leafy, stemmy, they're all built of, they're collections of cells, plant cells. One of the things that's different and unique about a plant cell compared with an animal cell is it has a cell wall. So this vertically arranged structure around the sides is called the cell wall. That's where all the fiber is in a plant cell. It's the external rim or rind around the cell. We talk about that as being cell wall. It's made up primarily of cellulose. Cellulose is the most abundant biomolecule on Earth. If you look out the window, you're looking at cellulose on the hillsides, whether it's forests, whether it's grasslands, you're looking at cellulose. Cellulose is by itself highly digestible by ruminants, but these cell walls also have a grid or a matrix of lignin particles or strands, and Gene likes to describe them to his students, and I think it's a great analogy, as the rebar, the reinforcing rod in a concrete wall. So if you think of the cell wall as the concrete or the blocks and maybe the mortar, but you think about the lignin as the rebar, the lignin is what gives plants their vertical structural strength so they don't fall over, but it also reduces the digestibility of the cellulose, because lignin by itself is indigestible. Now, I want you to think about also. Everything inside the cell wall is cell contents. We just lump it all together. It could be protein, it could be starch, it could be sugars, it could be various organic acids. But what's interesting about cell contents is regardless of the plant species, regardless of the maturity stage, regardless of the environmental conditions, cell contents are almost 100% digestible across the board. Doesn't matter whether it's straw, corn grain, loaf of bread, 98% digestible. So one of the concepts in modern forage testing, it's been out here since Pete Van Soest invented it in the 1960s, is if you can fractionate plant material into cell contents versus cell wall, well, you know right away the cell contents have essentially 100% digestibility, and you can set them aside. That's really easy to understand. What about the cell wall? We know that depending on the amount of lignin, which is a function of maturity, we know the cell wall can be high, medium, or low in digestibility. That's testing challenging. Okay, so here's the much more simplified drawing of the same thing, and here's where you need to correct your notes. Your notes, your handout, I think it's page 3, only shows these two inner circles. So pencil in the outer circle, but basically make the two outer circles the same diameter, the same size. So all this is representing, if you take a grass or a legume, I don't care, or a corn stalk or a fresh blade of grass from your lawn, let's think of it as a collection of cells. And so you can have a relatively thin cell wall, that's the stippled purple outer rim, or you can have a relatively thick cell wall. So obviously in this case on the right, the cell wall or fiber, let's call it neutral detergent fiber, that's the term that's used in forage testing labs, that's obviously going to be a higher percentage of the dry matter. So in this case, cell wall or NDF might be 40 or 50% of dry matter. In this case, it might be 20% of the dry matter, okay? The green stuff is cell contents, the highly digestible fraction that doesn't depend on maturity. So if we think about breaking plants into groups, young plants, leafy tissues, cool season grasses, C3 means cool season grasses, C4 means warm season grasses such as switchgrass or broom sedge, those would be warm season, corn would be a warm season. C3s would be orchard grass, tall fescue, Kentucky bluegrass, quackgrass, legume or grass. You can think about how they vary according to their classes just in terms of cell wall and cell contents. That's pretty easy, pretty powerful. Just a few more concepts before I go on. Forages, most of us, I think, understand we're raising forages primarily as a cost-effective source of energy. I think sometimes we overemphasize protein as the main reason we're raising a crop. It's really energy. Forages are less energy dense, though, and they vary so much more than concentrates, corn, barley, oilseeds. They vary so much more that we can't use book values for forages to make feeding decisions. They're too variable. Book values are just averages. Sometimes they're helpful averages, but they don't really get at the kinds of decisions Gene is going to lead you through. So the other thing to think about, fiber is actually a plus. We have to have minimum levels of fiber in ruminant diets. It's a requirement in terms of health and digestive function, and Gene, I think, will maybe talk about that a little bit more. Now, I want to throw in a qualification here, because when we talk about forage testing, we talk about nutritional values, sometimes we get mixed up with how to interpret those numbers in a grazing or a pasture setting. We can have pasture that's outstanding in composition, in energy, in minerals, in fiber, in digestibility, but if we have sparse, thin, short pastures where animals aren't getting adequate bites and they're not getting adequate daily intake to meet whatever our target level of performance is, quality becomes a rather less important part of the picture. If you look at the various factors that explain or predict daily animal performance, there's actually more weight assigned to intake, daily intake, than to quality. So sometimes it's easy to misinterpret a test showing high quality in your pastures if you're not paying attention to, do my animals actually have enough of a mouthful throughout the day to meet their requirements. Major sources of variation in forage quality, we've already talked about a number of these. Leaf proportion, leaf versus stem. Leafier materials are going to have lower fiber, higher cell contents. Maturity stage, I'll show you a couple diagrams here. And one of the tables in your handout, by the way, leads you through protein, fiber digestibility as you go through the maturity stages. I think that's on page 4, if I'm not mistaken. Species, we've talked about a bit already. Again, C3 grasses being the cool season species that we're so familiar with here. But a lot of you have C4 species, and another warm season grass, for example, would be crabgrass. A lot of us are paying more and more attention to crabgrass as a possibly beneficial component in our summer pastures. Couple more things I wanted to just mention here in terms of major sources of variation in forage quality. When we look at making hay, if we drag the hay drying process out over days, if we have particularly thick, heavy windrows, we don't spread them out widely enough to get them to dry down quickly, we can lose an awful lot of the energy in the cell contents through this process of respiration. Cut plants are still alive till they dry down enough. They're still living things, even though they've been cut off, laid on, back on the stubble. And so as long as they're alive, they're going to be consuming sugars, releasing water and carbon dioxide. And in that process, they burn up the most highly digestible, valuable part of the dry matter. Those plants aren't just losing water as they dry, they're losing high-value sugars. And so anything we can do to shorten and speed up that drying process, get hay from field fresh, wet condition to baleable dry condition, or to the condition we're going to ensile in, if we can shorten that from 3 or 4 days down to 1 or 2 days, we save the highest quality constituents in that dry matter. We preserve it in the bale or in the silage. So the other impact of that is if we did preserve that material, those highly digestible sugars, we now have more carbohydrates available for silage fermentation that will produce more of the acids, that will lower the pH of the silage, that will stabilize the silage, prevent the silage from being as subject to spoilage organisms. And so there are a lot of real positives that are associated with this retention of sugars in our forages. Okay, real quickly, this is review for virtually all of you. If we look at the maturity stages of grasses and legumes as we go through from vegetative, young, leafy, regrowing plants up to mature, stemmy, flowering or seed head forming plants, what's happening to dry matter production in terms of tons per acre? Well, obviously, that's going up and up and up. If you think about one cutting of grass or legume hay, you might get something like 2 tons in a single cutting, maybe even more than that if it's a really productive field. So we've got dry matter production here, but as the dry matter is increasing, and you can see an awful lot of that dry matter increase is from what part of the plant? I want you to at least compare the leaf number, the leaf content from here to here to here. Do you think leaves are changing much? Amount of leaves, if you stripped them off and dried them and weighed them, do you think there are many more leaves here than there are here than there are here? About the same. But what's contributed to this yield? If the leaves didn't change, what changed? The stem. We got more stem here than we did here, than we did here, and certainly than we did here. So think about an awful lot of what's going on as these forage plants mature is stem production. Now that's good for dry matter production. That's good for tonnage. That's good if we only need moderate quality forages. That's great. But if we need really high quality forages that are leafy, maybe we don't want to let this cutting go all the way out to mature stem stage. The other part of stem production is as they get more and more mature, the cell wall gets more and more lignin deposited, and so the cell wall becomes less and less digestible as the plants become more mature. Does that make sense? And so consequences of that, digestibility of the overall dry matter drops, of course. So here's the scale. I need my glasses to read the numbers, but it's going to be 80% down to 50% digestibility of the total dry matter. So we're losing digestibility with maturity. You all know that. But probably more importantly, Gene might have a different point of view on this, we're also losing intake potential. Okay? That's dropping partly because the forage passes through the animal more slowly as it's more mature. And so intake, again, that accounts for more of animal performance than digestibility does, but they're both going down with maturity. So decisions have to be made. Where do you want to be in this spectrum? And there's no one answer to that, of course. It depends on the system. Now, that's about all I'm going to say about composition. Let's switch to lab analysis, forage analysis. You have to define a lot. Same concepts that Lewis talked about, and I've gone into much more detail in the handout. So normally we think of a lot as a field with relatively uniform crop composition. It can have weeds in it and it can be a mixture of grasses and legumes as long as it's relatively consistent, cut on the same day, handled, harvested, preserved in the same manner. You think of that as a source or a lot, just like Lewis has you thinking about a representative soil sample. So if we can think about that, we can go in with a probe like the one here I'll let you look at later with a sharp cutting edge perpendicular to the shaft. Why does that matter? There are a number of hay coring probes out there on the marketplace that have a tapered tip like a syringe. Great. Well, if you go to the National Forage Testing Association website, which is an excellent educational resource, it will lead you through specifically how to do a representative hay probe core sample. But it'll also give you a listing of about 12 commercially available probes, and it'll evaluate the features that an effective probe needs to have. And NFTA will tell you an angled tip, like a syringe, will bias the sample by, let's see, I think it tends to scoot between the stems and it tends to capture more leaf than a square-ended tip. Things you wouldn't necessarily think about. So you can bias the sample just in terms of the shape of the tip, okay? So this is a great reference if you want to learn more about what kind of a probe should I buy. Take 20 or more cores from this lot. So it could be one per bale. It could be one from every 10 bales. It could be 2 or 3 from what you think of as the representative large bale, but make sure you get large bales so that you're spreading your samples out over. There's still going to be variation within this lot. Composite them together into a single sample. That's what goes to the lab. If you make it too large, though, be aware many forage testing labs won't necessarily grind the whole sample. If they get a grocery bag full, they'll probably subsample that. It's a huge mistake because it's coarse material and some of it's stem, some of it's leaf, some of it's powder. It's very, very difficult, if not impossible, for a forage testing lab to subsample that correctly. They need to grind the whole sample. So don't give them a sample that's too big. Sample splitting requires ground samples. A lot of us dream up these schemes of comparing labs. We've got 4, 5, 6 really good labs in the US, just top-notch labs. And we'd like to know which samples should we send our materials to in the future. So I'm going to split my sample into 3 or 4, and I'm going to send one to Dairy One in Ithaca, New York, and I'm going to send one to Cumberland Valley Analytical Services in Maryland, and I'm going to send one to Dairyland in Wisconsin. Well, guess what? We just sent them 3 different samples because we didn't grind the sample first. We did it in a coarse manner that came straight out of this probe, and you just can't do it. You've got to get the sample ground first. Any reputable forage testing lab will grind a sample for you. They'll want to do an analysis on it, but they'll also give you back this leftover bagful, and you can split that or ask them to split it and send that around to the other labs if you want to compare labs. Okay, so basic probe technique is go through the flakes on a bale. So here we've got a conventional small square bale, rectangular bale, and the flakes are going this way. So we're going to go through the end of the bale, between the strings, because there is actually a surprising gradient of leaves and stems within a bale, and you can bias the sample. So go in the end of the bale. Similarly with large bales, go through the layers, through the flakes. Again, NFTA, if you want to learn more about tip patterns, tip shapes, whether they should be smooth, whether they should be serrated. Here's an example of a tapered tip. Read what NFTA has to say about that. It's a wonderful probe. It's beautifully engineered. It's a very nicely designed and manufactured probe. NFTA has an opinion on it. You may agree or disagree with NFTA, but at least read their stuff. If you have a probe like this one that has a chamber on it, you can composite your 15 or 20 samples all together very conveniently into one canister, dump that into a bag, send that off to the lab. It's a lot easier than a probe. Some of the probes are just a T-handled push type that only take a single core, and with a ramrod you have to extract the core and then take another one and extract the core. These can be a lot faster and more convenient. I'm going to skip, I need to move on. I'm going to skip the background on detergent analysis. It basically is the means by which we get the cell wall versus the cell contents we talked about. It's done with boiling detergent solutions. It's basically fancy dish soap. If you put a ground dried sample into this boiling detergent solution, boil it for an hour, which is what we've done here. So here was the 1 gram of forage dry matter. If you boil it, you separate the cell contents from the fiber. Here we go. We're going to pour the solution off through a filter paper. What goes through the filter paper is the cell contents, the highly digestible fraction. What stays on top of the filter paper is neutral detergent fiber. That's the part that's partially digestible. Okay, and so to calculate neutral detergent fiber, it's just how much of this residual insoluble sample, what percentage of the original starting sample is that? So in this case, it's 50%. The other aspect to forage testing that's become much more commercially available than it was 10 and 20 years ago is using Rumen Fluid to actually run small digestibility bioassays in laboratory test tubes. And so there are ruminally fistulated or cannulated animals out there, a surprising number of them actually, that we get rumen fluid from. It's under very controlled conditions. They have to be fed in very specific manner, has to be gathered in a very organized way, but you can take rumen fluid that's full of these microorganisms that'll decompose fiber. It goes into a insulated, heated thermos flask. Oxygen is excluded, brought to the lab. It's filtered through filter paper, and you get a solution here that you can actually incubate dry ground forage samples in a test tube environment, simulating the rumen. It's done at live temperature under anaerobic conditions to exclude oxygen. And so we can get little mini digestibility assays from this kind of technique, and it's now affordable readily available from at least 10 labs in the US. Okay, and so that leads to some of the final energy estimates that we get in these forage test results. I'm going to try to wrap things up here. When you get forage test results, some people do their interpretations based on a single constituent like crude protein or like neutral detergent fiber. And if you think about historically at least, we've always thought of a reasonably good alfalfa hay as having, quote, 20, 30, 40 in composition. What's that mean? 20% crude protein, 30% acid detergent fiber, 40% neutral detergent fiber. Those are just numbers around in your head as kind of indexes of reasonably good forage, okay? And I'd probably add a 60 to the end of that. So 20, 30, 40, 60 or so. And the 60 would be this dry matter digestibility, or TDN, that we can get from these Rumen Fluid bioassays. And actually, if you look at, I think it's sample 6 on your list, the last page, if I'm not mistaken, sample 6 is essentially 20, 30, 40, 60, or 20, 30, 40, 63. So there's an example for you if you want to go look at it in the display. There's another approach. Instead of using any one of these constituents, we can add a number of them together and get a more comprehensive kind of an index. Those are called summative equations. I'm not going to go through the details, but they lead to a TDN or a dry matter digestibility, and I think Gene is going to lead you through those. I'm going to end with just a quick note about near-infrared reflectance. It's a technology that uses, it's a rapid, non-destructive, rapid, low-cost technique that uses light. You can irradiate a sample. It's light that's between visible light and heat. Infrared is heat. Well, near-infrared is invisible. It's just short of the heat spectrum. We can use this kind of a technology. We put a ground-dried sample in a small cell with a glass window, put it in this instrument. We can get a spectrum. And so this is just an absorbance spectrum at a number of different wavelengths in the near-infrared region of the light spectrum. And the amplitude of these absorbance peaks has to do with water, protein, and carbohydrate constituents in the forage. And so we can use this kind of information. I just got 3 samples that are on your list here. We've got a very low quality grass, BHY, a medium quality grass alfalfa, a high quality alfalfa. I've just put the spectra here, and you'll notice there's a flip-flop. So on this side of the spectrum, they're in this order. This side, they're in this order. There's a lot of complexity to a near-infrared spectrum, but we use very sophisticated software to interpret these patterns, and we can turn this into crude protein, digestibility, fiber, lignin, etc., at very reasonable cost. Choosing a forage test lab. I'm going to encourage everyone to go to the National Forage Testing Association site, NFTA, and look at the list of labs that belong to NFTA. They all have to pass proficiency tests in order to be listed there. That means they receive test samples, blind samples. I think it's 3 to 4 times a year. Do you know, Gene? 4 times a year. They receive samples that they don't know the composition of, that they have to analyze and return to the headquarters office, and their analyses, it's typically crude protein, ADF, and NDF, their analyses have to be within certain limits in order for them to even get a passing grade, and they can still get an A, B, C within passing grade. So you can look at which labs passed these tests and what was their grade. If you're using NIRC, the lab belongs to NIRC. Almost every lab that does NIRS testing belongs to that organization. I'm going to finish with request return of remaining ground sample if you want to send a sample to another lab. I think I better stop there so Gene's got enough time. Here's a typical test result. Again, we're comparing alfalfa with cool season grasses with pasture. I'll help you with these URLs if you want them later. We better go to Gene. And by the way, we do have, let me make a quick, I'm interested, completely aside from quality, I'm interested in anyone who may have information or may have bird's foot trefoil on your farm, or if you're aware of bird's foot trefoil that's been out there on the landscape for 10 or more years following a seeding. There are a number of us that are interested in collecting seed of persistent older stands of bird's foot trefoil that can just be patches, that can be scattered plants, that we could use in a variety improvement project to increase the persistence of bird's foot trefoil. I've got a few slips that I want to pass around on the dinner table that might remind you of this. But if any of you know of bird's foot trefoil that's surviving well following seedings that were done 10, 20, 30 years ago, please get in contact with me. I'd appreciate it.
While they're changing